rocking pneumonia and the boogie woogie flu. I know a lot of influenza songs, right? Hey, it inspires a lot of artists. If you want to listen to the rest of the song, there's the link. It will be on your PowerPoint, so you can listen to it on YouTube. It is quite catchy and might keep you going when the caffeine isn't. So, what are we going to do today? Well, we're going to talk about the antimicrobial. So, these are the antibiotics and more. A lot of these you had in the cells week, right? When we talked about cells, and I tied in certain structures that bacteria have that are unique, that make very good targets. I even talked about a few antivirals. I'm going to add a few more, and so we have a few new things, but there are some hopefully some topics that you remember that are just repetition, and this will help you to pull it all together for the big upcoming quiz. Okay. All right. So, what are the objectives? Well, of course, we're going to talk about the drugs. We're also going to talk about some of the hosts' defenses. So, I'm just going to cover the first line of defense. Okay. So, the big news, however, is that the big quiz is coming up, the big one. And this is over a lot. So, if you waited to the last minute to study, ouch. But this is what's on the quiz. So, the introduction to medical microbiology, cells, viruses, control, antimicrobials, and again, the host first line of defense coming up. Okay. All right. So, microbes, hosts, and chemotherapy. I have a little picture of a place, a pharmaceutical store where you can go in and pretty much get any kind of antimicrobial that you feel like purchasing. You may even know people like this. I know of a family who would always send certain relatives to Mexico every year, and they would literally buy garbage bags full of antimicrobials and drugs because they are cheaper down there. Let's face it, it's tough to buy drugs here in the United States. It's expensive. All medical care is. So I understand the need to bypass the costs here in the United States. However, do they know what they're doing? Maybe you know people like this. They just self-prescribe. They're not feeling good, so they just grab something off the shelf. Now, I know that antibiotics don't work on viruses and colds, right? And influenza. You know that. People have told you that. But do you really believe them? Do you? Do you sometimes just slip in an unused bottle of antibiotics, when you're not feeling well, just because you think perhaps it might work? I mean, what do they know? Well, this is why you take advanced courses at Clarkson College. It's just not being told what to do and doing it, although that's important, but it's understanding why. Okay, that's what higher learning really is all about. Okay, shout out to infection control nurses. What? Why? Like I mentioned before with Semmelweis, they have a very tough job. You're going to hear a little bit later on how we are having complete clinical failure of all antibiotics. So when patients come in, we can't treat them and they die. We have nothing. So really, the infection control nurses are very heroic. In fact, every time you wash your hands with soap and water, you are being heroic, probably saving lives that very day. Okay, we'll come back to that. Here's some terminology. We're going to talk about a lot of drugs that you have heard about even before you got into microbiology, medical micro. Some of these terms you heard in the cells, right? When we talked about prokaryotic cells. Okay, you would have heard of some of these drugs, but we're also going to talk about more kinds of drugs and the different classes that they belong to. So, where do these antimicrobial drugs come from? Where do they come from? Well, I'll tell you what. Microorganisms do not get along. Living creatures on a microscopic level do not get along. Well, they're living, and like all living creatures, there's competition. There's competition for resources and, for turf, and they don't want to share that. So, how are they going to fight one another off? Well, I have two dishes down below. And this was a plate of Staphylococcus aureus. See these little dots here? That represents bacterial colonies growing on a culture dish. But this is a particular bread mold growing. Notice where the bread mold is, there's no bacteria here. That is because the cells of the mold are manufacturing and secreting a particular drug called penicillin, sorry. And the mold itself is called penicillin. My bad. So you get this dead zone here. Well, it works both ways. This is a colony of bacteria, and they are fighting off mold. Sorry about I had the stupid link coming up. I shouldn't have that, but yeah, chemical warfare on a microscopic level. Pharmaceutical labs literally will go out and sample soil in fields trying to find antimicrobials. At least they used to, although there's no money in it now. So, why is that, you ask? I'm so glad you asked that. You know what? So, if your patient takes an antibiotic, how long are they taking that antibiotic? 7 to 10 days, right? Well, that's not a big return for our pharmaceutical companies. Now, if you're thinking, but there's always germs, there's always bacteria out there. But here's the deal. Bacteria replicate and mutate much faster than you can ever get FDA approval for a new drug. And to get a single drug approved costs billions, probably more dollars. And the drug company only has about 7 years before some other drug company then can steal the ideas. Well, it's not really stealing because the patent has run out. So the drug company has lost its exclusive rights. The other pharmaceutical companies will move in, have Me Too drugs. So, yeah, the drug company only has 7 years to recoup over $1 billion of investment, and the bacteria will always mutate out of it. Just so you know, we haven't had a new class of antibiotic for over 4 decades. 4 decades. But the drug companies are rich, you say. Yes, but they have shareholders. So where's the money going to be earned? Well, okay, let's go out and give patients, develop drugs for cholesterol, high blood pressure, asthma. Hey, we've got you there, and we have you for life. Maybe reflux. Okay? Yes, there's truth, isn't there? Okay, let's turn the clock back. Let's talk about the first discovery of the true antimicrobial. The one that started it all. This is penicillin. So penicillin was discovered by Alexander Fleming. He was a chemist. He was a bit of a slob, but he was a very good scientist. So he was working in the lab and he was trying desperately to find a cure for Staphylococcus aureus. And a lot of scientists of the day, this is the early 1900s, they were all working on this problem. They were using chemicals from even from the textile industry, dyes, things like that. So Alexander Fleming was busy at work and he had a little contamination in one of his culture dishes of Staph aureus. Luckily, he didn't throw everything out. He carefully examined everything and he made a huge discovery that he knew it would change medical history forever. He noticed that wherever this mold was growing, this bread mold, no bacteria grew. He knew the mold had to be manufacturing some antibacterial. The mold is called penicillium, and when he extracted the chemical, it was called penicillin. So he won the Nobel Prize in the early 1900s. And again, he discovered this contaminated plate with the bread mold Penicillium notatum growing on the plate. And from there, okay, he deduced that there was some chemical being released. There's this little zone of inhibition, no bacteria grew. He thought he had it. Eureka! Yay! Oh, sorry. Um, there is one little problem. Poor Alexander Fleming was a really bad lecturer, and nobody would listen to him. He really wasn't the person, though, that came up with the antibiotic that we take nowadays. Some people, they were from Australia, really. They got a hold of his information and they were also better at talking to people. So they developed the drug that is sort of the beginning of the penicillins that you know. So poor Alexander Fleming, he was very frustrated. But okay, so he wasn't very neat, and yes, he couldn't talk. But he was a good scientist in every other way. Okay, so let's ask a question. Let's stop for just a little quiz. Why do organisms such as penicillium bread mold make antibiotics? A, they manufacture many unique chemicals that attack other microbes. B, they win turf by chemical warfare. C, they don't like sharing nutrients with other organisms. Or D, all of the above. Of course, all of the above. And Alexander Fleming knew that. By the way, this is St. Mary's. This is the laboratory up here, and this is where he was working. This is Alexander Fleming's lab museum. It's still a mess. All of his things are still there. Yes, this is me. A friend shot this picture. We were actually on the street to go find a haggis sandwich shop in London. This is in London, by the way. Yeah, they sell haggis in London. And it just so happened it was around the corner from St. Mary's. And my friend said, "Hey, you want to see where Alexander Fleming discovered penicillin?" Yeah, I about dropped dead. Yeah, let's do that. All right. How about some terms? Okay. So, chemotherapy, when you usually hear that term, you're probably thinking of some kind of drug that is used to treat cancer. But chemotherapy, if you break that down, just means that it's some chemical that treats a disease. So, it's a treatment of disease with a chemical agent. Maybe if you use the word chemotherapeutic agent, it wouldn't become mistaken for chemotherapy used in the oncology department. But okay, this applies. All right, so antimicrobial chemotherapy. Some more terms. Anything that's antimicrobial will kills anti or microorganisms. You knew that. Antibacterials kill bacteria. All right. Antibiotics. Can these technically be interchanged? Can antibiotic be interchanged with antibacterial or antimicrobial? Not really. An antibiotic, by definition, is naturally made. So the bread mold penicillium manufactured and secreted penicillin. So on that petri dish, Alexander Fleming found an antibiotic. It's natural, a natural metabolic product of living cells that are detrimental or inhibitory to certain microbes. Okay, so antibiotics are natural. Now, on the other end of the spectrum, there are some antimicrobials out there that are synthetic antimicrobials. That means that they were manufactured completely in the laboratory. They're still organic, but they're not natural. Usually, scientists use computer models to figure this out, or trial and error, even better yet. Now, I do still use antibiotic for communication purposes, and you will probably need to do the same. Just like you might have to use the word bug instead of microorganism when talking to your patients. But again, an real antibiotic is natural. Now, one other term is the semi-synthetic antimicrobials or antibiotic. These are actually drugs that started out as the natural drug. So they started out as an antibiotic, they were natural. And the scientists said, oh, this is really a great chemical like penicillin, but it only kills a few different kinds of bacteria, or it's excreted very quickly, or bacteria develop resistance against it very quickly. So they take a little bit of the chemical and they tweak it. Okay, they tweak it a little bit and they make a semi synthetic antibiotic or a semi synthetic antimicrobial. And most of the drugs that you take and that your patients take are semi synthetics. Okay, some other terms. There are drugs out there that are narrow spectrum antibiotics. What does that mean? Well, that means that they only kill or inhibit certain kinds of bacteria, as opposed to those that are broad spectrum antibiotics, and they destroy a lot of different types of bacteria. Now, just because something is narrow spectrum, that doesn't tell you that they're killing only gram positives or only gram negatives. There are narrow spectrum that will only kill one or the other or just a few of each. Okay? Now, just because something is broad spectrum doesn't mean that everything is going to die. Remember our list in the control lecture. Some things are much harder to kill than other things. Okay? So, broad spectrum doesn't mean everything, it just means many different kinds. Okay. By the way, here are some broad spectrum antibiotics. Now, when you were listening to Mr. Seberg, you probably learned that there were some chemicals that were polar and some that were nonpolar. Some are larger and some are not very large. Now, what kind of drug do you think would do a good job getting through a membrane of phospholipids to get into the human cell, get into the bacterial cell, get deep into the tissue? Well, if you're thinking, maybe something that's not charged, something nonpolar, those phospholipids, they don't like those things. Maybe something sleek that looks like a race car. Well, like tetracyclines are very sleek. They get into a lot of tissue, into cells. Boy, they're very broad spectrum. Chloramphenicol, that to me looks like the kind of like a bit of a broken Starship Enterprise, goes everywhere. A very broad spectrum. I know. Shout out to you nerds. Hey, hey, high five. Okay, and then there are some that are still broad spectrum, but not that broad. So this is erythromycin. So we'll talk about these, okay? Antibiotics. Which one should I take? Let's just take all of them. I have a cold, and I think I'll just take them all just to make sure. Is that a good thing? All right, so let's start off by just stating something very clearly. We can kill every single pathogen out there. Oh, yes, we can. But what about the host? This is something that we used before penicillin. So this has arsenic as well as iron and strychnine. Strychnine is used as a rat poison. And this just shows you how much is in there. This is from Park Davis from Detroit, Michigan. Yeah, Park Davis, still kind of big. But if you're appalled by this, hey, they are chocolate coated. So, back off from Park Davis. But this would work. Yeah, if I consumed rat poison, I'm pretty sure every microorganism in my body would be dead, and so would I. So, yeah, by the way, this was used for syphilis, if you're wondering what was this prescribed for, and other things. So pretty much, a lot of infectious diseases got chemicals like this. So let's take a moment to talk about selective toxicity. Selective, that means, okay, it's only toxic to a few things, preferably to the pathogen. It's toxic only to the pathogen. So the drug is a poison, but it doesn't poison the human host cells. The drug doesn't target similar structures. So this is you, okay? You have many structures in here. Antibiotics, as you understand them, they are all poisons. They are designed to be poisons. But the trick is to poison bacteria and not you. So, yeah, selectively toxic. And just think you are made up of all of these cells, and if you consume these drugs, they're going to go through many different organs, especially, oh, kidneys and livers. Okay. Some of them, the broad spectrum that can really travel, get deep into tissue. Yeah, toxicity issues. Okay. All right. So, what is the ideal antimicrobial drug, you ask? I'm so glad you asked. So, a drug that has selective toxicity, it kills the germ, it kills the pathogen, and doesn't kill you. By the way, narrow spectrum is best. What? Why don't we just go in there and just start shooting? Okay, calm down. Calm down, Tex. Um, Narrow spectrum doesn't eliminate the good guys, right? Try to narrow it and try to find the one thing that's causing the issue and leave everybody else alone. So if the patient's having a gram-negative infection, then give the patient a drug that targets gram-positive bacteria. Give them, if you know the patient has staph, and 9 times out of 10, that's usually true, or strep, those are gram-positive bacteria. Okay. Remember, gram-positive stained purple got lots of outer peptidoglycan and are susceptible to penicillins. Sorry. But yeah, give them a penicillin. That'll kill the gram-positives, leave the gram-negatives alone. Good idea? Yeah. Okay, leaves the good guys behind, at least some of them. Try to develop an antimicrobial where the bacteria can't easily develop resistance against the bacterial genes. So here we have, let's say we have some bacteria in here. Somebody developed a mutation very quickly to our drug, and then that one divides and takes over. That's not a very good drug. By the way, methicillin, as in methicillin-resistant Staph aureus. Methicillin was a type of penicillin. The bacteria developed resistance against it very quickly. Vancomycin, okay, that drug, our last defense, it took bacteria about 3.5 decades before they could mutate 7 genes to become resistant against vanc, but they did. No undesirable side effects. So it'd be nice if you didn't break out in a rash, go into anaphylaxis, you didn't lose your liver or your kidneys, meaning you'd have to go into dialysis, no neurotoxicity, and it would be really great again if you didn't have to lose all of your normal microbiota. Because if you do, well, if the patient does, they're going to be on the toilet probably for quite a while and they're going to be sick. Okay. So, what do these drugs target? What are the mechanisms of drug actions? So here's a picture. We're going to start off with the cell wall targets. Why? Because those are the best and the most used. Those will include something called beta-lactams. What's it? Penicillins. More on that later. Vancomycin is in there too, a few others. We're going to talk once again about fluoroquinolones that attack DNA and RNA production. We're going to talk about sulfa drugs that attack folate production, cell wall membrane disruptors, and the big protein synthesis inhibitors, such as tetracycline, Z-Pak, streptomycin, all kinds of big stuff. So coming up here. All right, what are these beta-lactam groups, or these beta-lactam drugs? You might have heard a physician saying, well, yep, patient has a gram-positive infection. So you should think, staph or strep, maybe? Okay. And so, oh, we're going to prescribe a beta-lactam drug. And you walk into the patient's room and your poor patient's saying, what's a beta-lactam drug? And you say, it's okay. You heard of penicillin and ampicillin and things like that. It's just like that. Okay. So then they go, oh, okay, I've heard of that. All right, we're okay. So yeah, beta lactams is a huge group of favorite. All of the drugs in this group are called beta lactam drugs because they have a beta lactam ring. Look on the right-hand side. See these two chemical structures? All you have to know is that there's that ring in red. This ring is the beta lactam ring, and this is the chemical structure that all beta lactam drugs have. And this is the chemical structure that just, messes up PBP enzymes. Oh, there's that word again. The bacterial enzymes that make bacterial cell walls. This is the structure, okay, that blocks that PBP enzyme. Yeah, think about that for a minute. Blocks the PBP enzyme. That's what it targets. More on that in just a minute. Okay, so how am I going to remember which are beta lactam drugs? Okay, I'm going to try to help you remember a little bit. So you know the penicillins now. Penicillins, beta lactams, my favorites, except for the fact that I can't take them because of rashes, but and anaphylactic shock. But other than that, all penicillins pretty much end in cillins, like penicillin G, ampicillin, amoxicillin, methicillin, all of those. Now, the next group you may not have heard of, but you might hear a drug that begins with the word or begins with the prefix ceph. Ceph, those are cephalosporins. Usually ceph is in the name of the drug. Okay? So if ceph is there, you're like, oh, it's a cephalosporin, beta lactam, works like penicillin. So I can tell my patients, just like penicillin. Okay. Then there are the carbapenems. The what's its? Okay, they sound like M and M and M and M and M's. Carbapenems. Okay. When the doctor says carbapenem, you probably know that your patient is kind of in trouble. Okay. Not serious. Hopefully the carbapenems will be okay. But yeah, they might be in a little bit of trouble here. So doripenem. I see penem there. I think, that's a carbapenem. That's a beta lactam. All of these are going to mess with PBPs and therefore mess with cell wall production. Remember this? Okay, so here's the bacterial cell wall. Think about the bacteria that you guys are working on, okay? So if you have a gram-positive, and I can't remember, well, I don't think any of you really have drugs that really, that are going to be good on your particular organisms, but you still need to know this. Okay, so, but remember, all of your bacteria do have peptidoglycan cell walls, all of you. And you have, okay, unless you had something like walking pneumonia. So here are the NAG and NAM. This is the sugar of the glycan part, peptidoglycan. These are the peptide chains, the amino acid chains. Here's the enzyme, PBP, comes in and links them together, and then there it goes, okay? Now, penicillin, this thing, looks like a P for penicillin. Yeah, this will come in and mess up the PBP enzyme. When that happens, look how the cell wall is not put together. When the cell wall isn't put together, that's bad for bacteria, good for us. The PBP or the beta lactam drugs do this. All right, so beta lactam drugs. What are the target of beta lactam drugs? All beta lactam drugs. The penicillins, the cephalosporins, the carbapenems. The target is the PBP enzyme. That's the target. So what is the PBP enzyme? What's it supposed to do? It's supposed to link the peptide chains in peptidoglycan. That's what it's supposed to do. If it's blocked, then the peptide chains aren't linked by the PBP enzyme. Now, the bacterial cell will still try to divide. The DNA will try to replicate, but without a rigid cell wall to protect the cell will explode, or lyse is another fancy name for explode. Now, if a bacterial cell is exploding, that's bactericidal. It will kill the bacterial cell. But sometimes this won't work because sometimes bacteria aren't dividing. They're not making new cell walls. So the PBP isn't even there. The cell wall's already there, it's intact, and so it won't work. So this only works if the bacteria are actively growing. One of the things also with beta-lactam drugs, they're not great at getting inside infected cells. So if you have a white blood cell and you have something like if a bacterial cell's hiding in there, this isn't going to go in and get it. All right, it can go into the tissue, but it can't get into the cells. So you wouldn't use any of the beta-lactam drugs for Rocky Mountain spotted fever because those get into cells. Those are intracellular bacteria. Okay? But pound for pound, the beta-lactam drugs are the best drugs to use for selective toxicity. And the penicillins, wow, if you can do a penicillin, that's best of all. Okay, hint. Just know the specific names of the antimicrobials that are in red font for the quizzes. You need to know the groups, and you need to know that this antibiotic belongs in that group, but there's a lot of drugs out there. And to be honest, even the physicians don't know every single drug name, and drug companies will put their own little name to it. But if you look at the ingredients, it'll have the basic antibiotic name. Yes, I mean antimicrobial name, okay? So just the ones in red, so don't panic too much. And some of these you've heard of. So hopefully it's review. Okay, so if you can remember during your quizzes, okay, we talked about that at the beginning. So that must be some kind of drug that attacks cell wall production, cell wall synthesis. So yeah, that's a cell wall synthesis inhibitor. And even the first one she's talked about were beta-lactam drugs. Okay, so let's start talking about those. First one, penicillins. And I'm sorry, it's a small font. I couldn't get it all on the slide otherwise. So, but I'll take it a little bit at a time. I could have broken it up into many slides, but I thought this was better if you were looking at the PowerPoints. You could compare. So, types of penicillins, the cillins. Well, first of all, know that they are all well, the first ones were pretty narrow spectrum. Okay. So the first ones mostly treated gram-positive bacteria like Staphylococcus aureus, but it also would take out a few weird gram-negatives like Neisseria gonorrhea, well, at the beginning. So penicillin G, that was the original form from the mold. That was the first drug, okay? So the penicillin G, by the way, and you don't need to know this for the test, but there's two penicillins. There's penicillin G, penicillin V. And which one is which? I always think about penicillin G as the injectable, and I'd say, oh, gee, that hurts. Okay. As opposed to oral, which is the penicillin V. V is in victory. Okay. G is okay. So penicillin G, like I said, it's very narrow in spectrum. So, the drug companies, they said, can we tweak this a little bit and make it a little broader spectrum and maybe make some extended spectrum penicillin drugs? Okay. So they're still penicillins, but not penicillin G or V. They're a little more broader, a little more extended in their spectrum. So let's talk about those broad and extended spectrum penicillins. Like I said, they were developed to kill more types of bacteria. That would include ampicillin. Ampicillin, you don't need to know this, but you probably have seen this in hospitals. This is the injectable kind of penicillin. Okay. And it's semi-synthetic. Like I said, they tweaked the natural antibiotic and made it a semi-synthetic ampicillin. And ampicillin is actually pretty broad, but again, it's not going to work if the bacteria climbed into one of your cells. So, yikes. Methicillin. All right, we learned about this earlier. This didn't stay on the market for very long. It was developed to be broader spectrum. However, the bacteria very quickly mutated and found genes to resist methicillin. And so, yeah, and these are semi-synthetic. Um, and again, the resistance gene gave bacteria resistance to the drug very quickly. It didn't stay on the market for long. But this is where we get the name MRSA, which stands for methicillin resistant Staphylococcus aureus. Really that name is so out of date, and I wish they would just change this because nobody prescribes methicillin anymore. They just don't, as far as I know, because the bacteria are like, that was so yesterday. So really, the M should stand for multi-resistant. Okay, so instead of methicillin-resistant Staph aureus, think of it more to stand for multiple resistant Staph aureus because Staph aureus not only learn to be resistant to this drug, but usually when they have the drug to be resistant to one penicillin, they're probably resistant to all or most of the penicillins. Yeah, they get resistance to them all. Clever little mutators that bacteria are. And probably not even just resistance to the penicillins. MRSA is probably pretty resistant to all the beta-lactam drugs by the time, but when they come upon a MRSA, they have to go out of the beta-lactam group and find something else. There's also something that you might hear about in the hospital. It's called piperacillin. You won't see this prescribed normally because, well, this is only kept within the hospital. We only give this to patients that really, really, really need it. We don't want it out there because once we release it to the public, they will abuse it. I'm sorry, it's true. Uh, and then the bacteria will have more opportunities to learn to mutate against it. Now, understand mutation is not something that they do to respond to antibiotics. The antibiotics just keep killing off everything but the resistant form. The mutations in these bacteria are random, accidental, but because they replicate so much, there are a lot of mutants, and somebody somewhere is going to accidentally get the right mutation and be resistant. More on that later. So again, piperacillin only in hospitals. What about the problems with penicillins? Well, yeah, with me, I can go into anaphylactic shock. Yeah. We'll talk about why this happens. It's not the drug, it's actually the host. It's you, it's me. My immune system thinks that penicillins are the worst thing since Ebola. Seriously, penicillin gets into my body and my body freaks. I go into such an inflammatory response. I go into anaphylaxis and I'll probably die unless there's a nice EpiPen handy. Okay? It's not the drug, it's the patient. There are a few other rare things that can happen with penicillins, especially if you directly inject it into a nerve. That can be bad. Okay? And again, the bacteria do develop resistance genes that make them resistant to these drugs. But I wanted to show you on the right-hand side, here's bacteria growing normally. But again, here are bacteria growing in the presence of penicillin, or really any beta-lactam drug. So the cell walls are not being manufactured. Water rushes in, the bacterial cells die. This is bactericidal in a big way. Okay, so more penicillins. I just want to mention amoxicillin is a special kind of ampicillin. They added a little chemical in there that is an anti-resistance chemical. The chemical is called clavulanic acid. I don't think I'm going to have you know that for the test, but it's the if you know of you like probably you know amoxicillin. If you have kids or know of kids, you know they have a lot of they take a lot of amoxicillin for earaches and if there's a human bite. And then once that kid or patient takes amoxicillin, they get the Hershey squirts. Yeah, she said Hershey squirts. Okay, severe diarrhea. It's not the ampicillin, it is actually the other drug, clavulanic acid. It just makes you sick. Okay. All right. What about that second beta-lactam group? And again, they have that beta-lactam ring. So the second group is the cephalosporins. And again, they started out as a natural antibiotic from fungi, but also some bacteria made these. This is a huge group and they are very broad spectrum. But again, you don't really give these to patients that have intracellular bacteria. So they worked pretty well until the bacteria got a hold of a resistant gene. Basically, they somehow got these genes randomly. They don't choose. You don't get to choose your genetic traits. Neither do they. But yeah, the genes made them resistant. Cephalosporins can't come in generations, which is actually a lie. Drug companies just use this for marketing. They're unlike penicillins, they didn't develop new generations for broader spectrum use. It's nonsense. But the first generation, okay, is cephalozoan. Second, cefuroxime. Notice these are not in red, so you don't need to know these. But in the third generation, cephalexin or K-flex. Gosh, that sounds like cephalosporin to me. Ceftriaxone, ceftazidime, and cefepime are also cephalosporins. Okay, so again, when you think cephalosporin, think, oh, those kind of work like penicillins, but they're broader spectrum. Okay. What else should you be thinking about with cephalosporins? Well, here's the deal. I can't take cephalosporins. And if you or your patient is allergic to penicillins, chances are they can't take the cephalosporin either. This is still a beta-lactam drug, looks a lot like penicillin, comes from natural fungi or bacteria. And you know what? A lot of patients will also develop allergic reactions to cephalosporin. Same structure means it can be the same type of allergen for your patient. So before I continue with this, I once tried this before I became less stupid. I knew better, but I still did it. But here's what you really should do. First of all, the pharmacy would never give you a cephalosporin if you were allergic to penicillins. They shouldn't. There should be a red flag. But if for whatever reason they need to do this, the pharmacist might suggest the following. So you get it in a tablet so you can cut it. You cut it in half. You cut it in half again. So you have a fourth of the tiniest little dosage tablet you can get a hold of. You take that little pill and you hang out with somebody, a medical person who has an EpiPen. Okay? If you're going to have a severe reaction, it will happen within the first 30 minutes. So do not leave the health professional. And maybe they might even need a tube just in case. But that's the best way to approach this. I did it and took none of that advice. I was an idiot. Okay. Everybody has moments, you guys. So I learned, though. I did learn. I did learn. It can happen. So, okay. And also, bacteria did develop resistant genes. So resistance genes made the bacteria resistant from these drugs. So didn't work anymore. What's our third beta-lactam drug? These are the ones that sound like M&Ms. Carbapenems. They are still beta-lactams. Look at that sleek structure. It's not charged. It is, you don't need to know this, but it's called a zwitterion. Basically, this thing really can move and it can get into tissue very well. Again, originally it was from a natural product, okay, from these kinds of bacteria here. Pretty darn broad spectrum. We do not want to prescribe these drugs, though, unless we absolutely have to, because we have bacteria out there that don't respond to other things, but they will respond to carbapenems. For example, if Pseudomonas aeruginosa, which is very gram-negative, is resistant against all the other beta-lactam drugs, we can give a carbapenem to the patient. Also, if the patient has enterococcus, like Enterococcus faecium, Enterococcus, basically we'll just stay with Enterococcus. I'll talk about, you're thinking, I've never heard of Enterococcus before. Bacterial, yeah. Coccus shaped, yeah. Have you heard of VRE? VRE? Okay, so the E in VRE stands for Enterococcus. More on that in a little bit. So we use these drugs against Enterococcus and Pseudomonas aeruginosa. If you're wondering what this picture is over here, ever been in a hot tub, come out with a hot tub rash? I hate to spoil all your fun, but listen, never get into a public or a hotel hot tub without a huge jug of Clorox. Okay. Yeah, you need to run Clorox or some kind of hypochlorite um, chemical through those pipes, and you let it set because Pseudomonas aeruginosa is very good at forming biofilms. And also, it is a master gram-negative bacterium when it comes to forming mutations against drugs. A master. So you're going to have a lot of patients with Pseudomonas in their lungs, especially if your patient has cystic fibrosis. So the physician will probably prescribe a carbapenem, right, carbapenem, to treat that Pseudomonas. And even then, it might not work. They might have to hit it with another drug. We call those fluoroquinolones, right? The FQs, fluoroquinolones. So might have to hit it with two totally different kinds of antibiotics because this is bad. This will kill your patient. Okay? This isn't deadly, but it certainly is going to make you uncomfortable. So, just like all the beta lactam groups, it inhibits cell wall synthesis. This is our last really good option to treat gram-negative infections. When this does not work, this is really bad. When you have a gram-negative bacterium like Pseudomonas aeruginosa or something called Klebsiella pneumonia, and they've tried everything, including the carbapenems, and if that isn't working, bad, 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 bad. They will then have to move to such toxic drugs that the patient may end up on dialysis afterwards, just from the drug, from the treatment. We would have to move to drugs that even stopped prescribing, took them off the shelf. And now, because of antibiotic resistance, they have to come back on the shelf. I'll talk about those a little bit later on. Those will be the membrane disruptors. Understand if a bacterial organism has has a resistance gene against one antibiotic, it probably has multiple resistance. So many genes for multiple resistance, and nothing is going to work. And this is when you have a superbug. Okay? This is where you have full clinical failure. And so, yeah, this is serious. And moving on. Okay. And again, we think of MRSA. MRSA is resistant Staph aureus. But remember, Staph aureus is a gram-positive. We have other things to treat Staph aureus with other than the penicillins. We do. We have a lot of things. So it's now not always. There is clinical failure of MRSA. Don't get me wrong. MRSA can have a lot of genes that can make deadly toxins, and the patient may not survive by the time we figure out which antibiotic will work. But again, the gram negatives, when they are resistant, this is terrible. The LA Times came up with an article about something called KPC. This is a particular gram negative bacterium called Klebsiella pneumoniae, and it was making an enzyme that knocked out the carbapenems. So we say that this is a Klebsiella pneumonia carbapenemase producer. Basically, if you see KPC in the clinic, in the hospital, think bad. A lot of these come from countries that do not regulate the use of antibiotics. So lots of people taking antibiotics. Antibiotics end up in the food. They end up in the water, in the sewage, and the bacteria have this wonderful little bath, this slurry of all kinds of antibiotics. And the antibiotics will start selecting out the weak bacteria and select for the strongest, the superbugs. This is particularly bad if you have a patient that has done a little medical tourism. You know, they went to another country to get a surgical procedure that was too expensive here in the United States. But in that country, there's a lot of abuse of antibiotics, and therefore the bacteria there are antibiotic resistant. There's a and by the way, India, that wonderful country with the best food, Of course, there's Bollywood and the most incredible fabrics in the world. Unfortunately, they're not good at regulating antibiotic use. And there's a lot of people that go there for medical tourism reasons. And I would love to see India, but it scares me. I really don't want to be colonized by resistant bacteria. I will feel fine, but if the resistant forms take over, and if I ever do need an antibiotic like a carbapenem, it won't work. Understand, though, this is a big problem everywhere. And again, if you see KPC or another one that I'm going to talk about, the New Delhi strain, you will notice the physicians freaking out and they will be calling every ID physician they can get their hands on, and the CDC will be called in. This will be bad. Okay, let's leave the beta lactam drugs. Okay, are there other ways to inhibit cell wall synthesis? Well, yes, absolutely. When you talk about the beta lactam drugs like penicillin, it only stops one enzyme that does one little tiny job. That's all it does. It just links together the peptidoglycan cell wall. Are there other steps that you can inhibit or stop? Absolutely. So bacitracin does that very thing. But again, it is not a beta lactam drug. It does not mess with PBP at all. Its target is a little lipid carrier molecule inside the bacterial cell. So what this little lipid carrier molecule, think of like a little carrier, um, like a UPS carry person or UPS employee, it carries bits and pieces of the cell wall to where the cell wall is being made. Okay, if you could block that little carrier, that little, um, like our UPS carrier, then the cell wall again can't get all of its subunits. So that will block cell wall synthesis. Now, bacitracin is topical only. It is over the counter, but it is topical. Why? Because the little devil is very toxic, too toxic for oral use. And because of that, really, there's not a lot of resistance against this, but it only is going to work for like a skin infection. But let me tell you, do not take any drug unless you absolutely need it. For the most part, soap and water cover the wound lightly with bandage. Plain and simple. Okay. Okay. How about another cell wall synthesis inhibitor? How about the last one? And actually, this one sort of is. This is our last defense, or at least it was, against resistant Staph aureus and resistant gram positives. I am talking about the big vancomycin. Like I said, not a beta lactam. Not at all. This is a massive molecule. Massive. Over here, all right, this is the chemical structure. No, you don't have to know this, but I sort of drew this little image to show you that this is Vanc in kind of a purpley blue, okay? Vanc, even though this is, the last effort, the last big gun for resistant Staph aureus, it's very, very narrow spectrum. It only attacks gram positives. But that's okay. Staphylococcus aureus is a gram positive, so this works. Also works for resistant strep, okay? Now, the target is a little weird here. So let's look at the picture up here. So here we have the cell wall peptidoglycan here, the glycan bars in green and kind of an off green. Here are the peptide chains, again, peptidoglycan, right? And the chains link down. Over here, of course, we have our little PBP enzyme that's supposed to link them together. What are these big blue things? I always think it looks a little bit like chewing gum, but it's a little more specific than that. So vancomycin will come in and latch on to the end of the peptide chains. These are actually alanine amino acids, a form of alanine. So it gums up the peptide chains. Okay, look down here. See, this is vancomycin. It gums up where these things are supposed to be linked. Well, then, again, essentially, PBP can't do its job. So the consequences are pretty much the same. So The vancomycin attaches to the peptide or the peptidoglycan peptide links. All right. It attaches to them and blocks the PBP enzyme. But again, it's not a beta lactam drug. So it isn't really considered a competitive inhibitor of PBP. So it's not beta lactam. Okay. Although the consequences to the bacterial cell are still the same, you're still not making a really good sturdy cell wall, so the cell could easily rupture. The problem with vancomycin, and there are several, number one, it's extremely, well, not only is it really expensive, but it's very toxic to the patient. Look at how big this is. If you are going, so if you do have a patient who has MRSA, you do go for vancomycin. Also, vancomycin is useful for treating C. diff. C. diff, Clostridium difficile, is a gram-positive bacterium, so vanc is used for that as well. You'll see it used for both. Resistant Staph aureus. Again, normally the physician's going to give some kind of penicillin or cephalosporin or oh, maybe a carbapenem, probably not. But if none of those are working, you give them vanc. If a patient has C. diff, you give them vanc, and this is what it does. So, okay, so again, not a beta lactam drug. The toxicity issue also has to do with the fact that we're pretty sure vancomycin messes with our membranes as well. I think there's some membrane problems when we take this in. Vancomycin, because it's so big, it will stay in the gut to kill C. diff that's in the gut. It will stay within the gut and won't get out. So that's one good thing. But if you have Staph aureus, like in the bone marrow, then this has to be injected. An oral can't get to the site where Staph aureus is infecting, okay, and causing disease. So, yeah, problem there. Now, also, we used to use vancomycin for enterococcus. Remember I said that? So that worked for a while until, well, enterococcus, which are types of bacteria that naturally live in your gut, they developed resistance against vanc. And so then we call those vancomycin-resistant enterococci or enterococcus. More about VRE later when we talk about resistance. And again, this shows the target is actually the peptidoglycan peptide chain. So red, red, green, red, red, green. They kind of fit right over the two reds. Okay, the two last little amino acids here. And just like chewing gum. This is another sort of interesting and startling problem with vancomycin. Sometimes if your patient is infused, so it's the IV kind, and they are infused too rapidly with vancomycin, they will get red man syndrome. I will tell you this picture doesn't do this justice. Patients look red. I mean, more like the red font here. And this will occur in less than 60 minutes. So this is, sometimes vancomycin is not prepared properly, but again, if it's fast infusion, this can happen. There are other drugs. Some of the Cipro can do this. Amphotericin B we'll talk about, and the others we're not going to talk about at all. But other drugs will do this. So if your patient's bright red, it's red man syndrome probably, and that's probably the issue. But what about the protein synthesis inhibitors? Some of you have as your favorite antimicrobial antibiotic protein synthesis inhibitors, right? Why? Well, okay, let's say your patient is someone like me. And if they take a beta lactam drug, they'll go into anaphylaxis. And why not vanc? Because that's pretty toxic, right? And expensive. We don't want that. And there are other drugs, but again, pretty toxic. So, yeah, and the protein synthesis inhibitors, a lot of them are good at getting intracellular. So, if I have Rocky Mountain spotted fever, I've got bacteria hiding in my blood vessel cells. I need something that can get in there and go get them. So that's another reason why we sometimes switch to protein synthesis inhibitors. So understand though, most of these medications that inhibit protein synthesis are going to target transcription or translation. Again, here is what happens at translation, right? And this is what you went over the most, what we went over in the cell unit, or the cells, when we talked about the cells, that lecture. So, most protein synthesis inhibiting drugs, by the way, are bacteriostatic, not bactericidal. What? Okay, so most of these drugs are going to only stop bacteria from replicating and not kill them. Why would we want that? Sometimes you don't need to kill the bacteria. Sometimes you just need to stop them from replicating so the immune cells can come in and num 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 munch them up by phagocytosis. Okay? And that might be a little gentler on your normal microbiota as well. All right? But there are some that are bactericidal. I will talk about those. All right. Now, all cells, if you remember, synthesize or make proteins. If you remember, the site of protein synthesis is the ribosome. Yeah, the ribosome. Ribosomes make proteins. You remember that. Here's our image. So here's our messenger RNA. Here is the ribosome about ready to be constructed around the start codon of the messenger RNA. It's going to translate that gene and make a strand of amino acids, but transfer RNA has to bring in the amino acids one at a time. And okay, now keep in mind, right, that prokaryotes do have unique ribosomes. They are 70S ribosomes. They have a large subunit of 50S and a 30S smaller subunit, right? As opposed to eukaryotes, they have ribosomes attached to those rough endoplasmic reticuli, right? The rough endoplasmic reticulum, right? Okay, those ribosomes that we have out there are 80S, so they're bigger and they have a 60S larger portion and a 40S smaller portion. However, there's something to consider. If you're thinking, yeah, no problem. So any drug that blocks this ribosome will go after prokaryotes only, right? So because drug companies they can actually design their drugs to go after the unique 70S ribosome, leaving our cells alone because we have 80S ribosomes, right? Yay! Well, okay. Little side note, what fancy people say, caveat, little caveat to this. In your cell, you have mitochondria, right? That's the powerhouse of each of your cells, like your white blood cell, your heart cell, your lung cell, kidney, bladder, all those. Okay. The mitochondria, the powerhouse of your cells, have their own ribosomes, right? And they are 70S ribosomes, like what bacteria have. So here's the problem. If you get a protein synthesis inhibitor that goes after a 70S ribosome, and it is really, really, really good at getting in through cell membranes, and let's say it's going after an intracellular bacterium, like the one that causes Rocky Mountain spotted fever. What happens if it keeps going and gets into the mitochondrion and knocks out their ribosomes? Yeah, there are toxicity issues with some of these, not all, but some of these. So, selective toxicity of some of these antibiotics can be pretty hard and not very good, I should say. And whether the drug is selectively toxic um, depends on several factors, some of which I can get into, some of which I can't. But remember, every antibiotic, all of those cell wall synthesis inhibitors, as well as the protein synthesis inhibitors, these are all poisons designed to knock out something in a cell. But we want to knock out their things, not our things. It becomes more difficult now. Well, Miss Eden, wait a minute, stop. What about like the penicillins? Remember, we don't have PBP enzymes because we don't have cell walls. So that is why penicillins, pound for pound, are the best. because the selective toxicity is really good with those, not as good with some of these. All right, so let's put these in groups. I know I have the little picture of the ribosome to kind of remind you of what these drugs, so this drug needs to attack the bacterial ribosome here. How? Well, the aminoglycosides, these are the rarest of the groups, but they are very toxic. And they, because of some of their toxicity, they are bactericidal. Remember I said protein synthesis inhibitors tend not to be bactericidal, but these are, yeah, real toxic drugs. They're broad spectrum. If you need them, you need to take them. Hopefully, no physician will prescribe these unless you really need them. Like I said, they attack the ribosome, the 50S portion. You don't need to know this unless maybe you're doing your paper on the aminoglycosides. Okay, and take a look at this, by the way. Sleek, very race car-like. This can get in through tissue and into membranes. So again, that also makes it potentially toxic to us. The two drugs include streptomycin and gentamicin. Gentamicin is the only one in red, but if you see the mycin, you should think, aminoglycoside. Oh dear. Okay, so it's bactericidal, but this could be ototoxic and nephrotoxic. Okay. I'll come back to that. Years ago, I was at the Travelers Clinic at the VA Medical Center, or excuse me, no, at sorry, at the Douglas County Health Department. I was going to get something for malaria, and because I was going to down to Ecuador, and I had to do a whole bunch of shots and things like that. And there was a really wonderful gentleman next to me, and he was a World War II vet. And he was so cool, and I love talking to vets, and I hope you do too. They love kind of revisiting thing. And we were just talking about where we were going, and he said, "Oh, I can't hear you. You have to speak into this ear." I went, "Okay." He says, "Yeah, I was in the army in World War II, and they prescribed, these antibiotics, and I couldn't hear afterwards." I said, "Okay, gentamicin?" He said, "Yeah." And I said, "Yeah. It is ototoxic. Okay, I didn't say that. I said it can make you deaf, is what it can do, because it's neurological. So it is ototoxic, attacks people's hearing, so you can take an antibiotic and end up deaf. It's also nephrotoxic. You can take the antibiotic and then need dialysis because it has kidney toxicity. Also, you wouldn't prescribe gentamicin for everybody and everything because, all right, not only does it have some nasty toxicity issues, but it's for aerobic bacteria only. If it's necessary, though, your patient is going to have to be given one of these drugs. Sometimes you don't have anything else to fall back on, okay? But hopefully you don't get an aminoglycoside like streptomycin or gentamicin early in life. If you have to take them, save it for later. Why? Because if you take it earlier in life, you may be okay and not lose your hearing or your kidneys. But because these drugs can remain in your fat, in your tissue, later on in life, it can accumulate and produce cumulative toxicity. And the second time you try that drug, maybe you're in your 40s or 50s, that's when you have to go on dialysis. Okay? Serious drugs. Tetracyclines, yay! One of the most widely used, one of two, and one of the most abused. So let's talk about those. This does look like a little race car, and it's not very charged. So yes, this gets deep into tissue, inside cells, goes after intracellular bacteria. Oh gosh, it'll do a lot of things, okay? Also used for treating things like syphilis and Lyme disease sometimes, okay? So tetracyclines, this is the tetracycline group. Sometimes you'll see the label and it will say tetracycline if it's generic, but it might also say doxycycline or minocycline. Doxycycline you need to know is a tetracycline. What does this one do? Well, still, it's stopping protein synthesis by stopping the messenger RNA translation into proteins, but it's actually blocking transfer RNA. Oh, what can go wrong with that? Well, we have RNA, they have RNA. But it does. It blocks the translation of proteins. Very broad spectrum. As I said, it will get in and kill intracellular bacteria. So when your patient has bacteria hiding inside their blood cells or inside the blood vessel cells, like Rocky Mountain spotted fever, there you go. It's also prescribed for acne. Few problems. Okay, number one. Sorry that you came up all at once, but it causes hormone disruption, especially bad in a woman who is on birth control and who is not in the mood to get pregnant. Okay, and they're relying on their birth control pill. Well, this is an endocrine disruptor, and therefore your patient can become pregnant if they're relying strictly on birth control pills. Okay, that's a big deal. What else? It immunosuppresses your patient. Some patients really have to watch, their immune response and get blood drawn because it will immunosuppress your patient. And so you can be on tetracyclines and get infections. Oh, joy. Also, if you have anemia, well, okay, iron does bind to the tetracyclines and therefore it doesn't get to where it's going and it can cause anemia. It concentrates in bones and teeth. So we never give tetracyclines to a pregnant woman because it hurts the developing fetus. And also, we don't give it to young children either because, well, if they haven't gotten their permanent teeth in, they will have the tetracyclines deposit in the teeth, and you get this discoloration. By the way, these teeth will be fluorescent under black light or blue light. Most of these patients, I had a nursing student once, and her poor thing, she never smiled. And when she was giving her presentation to the class, she let us know she was talking about tetracyclines as an option and why we don't take it anymore. We don't give it to like a young child that hasn't had their permanent teeth yet. And then she smiled, and her teeth were worse than this. The only thing you can do, by the way, is cap the permanent teeth when they come in. That's all you can do. One final thing. Okay. Tetracyclines cause photosensitivity. Why is there KFC here? And is it time for a chicken break? Well, okay. I'm going to talk about Kentucky Fried Chicken here, but understand this relates to most all chickens that are raised with antibiotics, and that's everything that isn't organic. So you can go to your favorite grocery store, and this is going to be true when you buy that chicken. It can be true in all of your meat. But let me tell you, um, yeah, okay, this is true for other things. I'm not just picking on Kentucky Fried Chicken. But okay, so here's the story. Dear friend of mine who received his PhD at Creighton was doing his grad work in the research laboratory next to mine, and he had a horrible rash. And he was out running. He was a runner, pretty healthy. But he was a Creighton student, so he he was stressed out, didn't eat well. And he had a horrible rash. And he asked one of the attending physicians that was going on. He was studying to become an ID physician, and he says, I've got this rash, doc. What's going on? And the doctor said, well, okay, are you taking an antibiotic like tetracycline? And he looked at him like, please. This is his thing. He's a microbiologist. In fact, he went on to become an opinion leader on antibiotics, especially with Pseudomonas aeruginosa treatment. But, he's like, no, I'm not taking an antibiotic. And with a smart alecky response. And the physician replied with okay, are you eating Kentucky Fried Chicken? Of course, that stopped him in his tracks because, well, okay, yes, Creighton student equals stressed equals eats badly, and he loves Kentucky Fried Chicken. So because tetracyclines are very commonly put into the feed of our livestock. Okay, you get tetracycline when you get the meat, when you get the food. Um, so yeah, I bet you didn't know you got free antibiotics with with your food. But yeah, and nothing the way that birds, chickens, turkeys are raised, they have to get a lot of antibiotics. So yeah, they get more antibiotics than most of the other animals. They will, by the way, feed other antibiotics to animals, not just tetracyclines. Antibiotics will make an animal fat. They know this. Okay. Europeans have stopped putting antibiotics naturally in their feed. So just so you know, they know something we know, we just don't want to stop because we've always done it that way. All right, let's talk about another protein synthesis inhibitor. This one is chloramphenicol. Chloramphenicol. We're just going to make it its own group, okay? So this one targets, again, if you have to talk about chloramphenicol, it attacks this guy or this guy, the 50S portion of the ribosome. Extremely broad spectrum. Looks like, again, the Starship Enterprise. Looks familiar? Yeah. And it goes everywhere. Yeah. Which means there's problems. It may get in and hurt mitochondrial ribosomes too. Consequences, okay, and there's probably other reasons we don't understand, but okay, vomiting, green stools, gray skin. It can cause gray baby syndrome, like that, which is, of course, very sad. This drug is what we call idiopathic. I don't think I have that for the test, but listen, idiopathic means you don't know when it will strike. Okay, so does it cause problems in patients that are young? I don't know. Patients that are old? We don't know. Patients that are taking other drugs, they have some kind of health problem? Nope. We never know when it will strike and be deadly. So this drug is toxic to the bone marrow. This is where all your blood cells are made. What could go wrong? Well, yeah, aplastic anemia can cause death in 1 in 20,000 patients treated. Okay. So here's a good story. So this is a case study I learned actually from an infectious diseases symposium some years back, but it was also repeated to me by Dr. Ken Thompson from Creighton University. And he said, so there was a patient that well, there was a young girl who had, they suspected Rocky Mountain spotted fever. They couldn't give her tetracycline, so they gave her chloramphenicol. She felt better and stopped taking her antibiotics, and then they put what was rest in the medicine cabinet. Okay, it didn't mean that all the bacteria were killed. She stopped having symptoms, but she stopped anyway. So Dad got of all things, a cold virus and thought, I'm just going to take an antibiotic. And yeah, there's some sitting here in the medicine cabinet. Okay, he had one and he died. Okay, this is serious. So this is why we don't use and, misuse antibiotics. Okay, one last story. China sometimes isn't really good about monitoring and regulating their antimicrobials either. And some years ago, some of their bees were dying, and the beekeepers were concerned because, of course, there's a lot of honey and bee products made from these bees, and they ship all over the world. So they thought, okay, well, let's figure out what it is. It was a bacterial infection that was causing disease in the honeybees. So let's see which antibiotic is good and broad spectrum. Yeah, you got it. They sprayed the hives with chloramphenicol. It was in the honey, it was in the bee products, the other products that like royal jelly, and it was sent throughout China and it wasn't imported here, thank goodness, but it was imported in other countries, including Europe. So take a look. Idiopathic, toxic to bone marrow. Look at some of these symptoms. I hope this is making an impact. You all will probably have to be very good at educating your patients to keep them from making horrible, even life-threatening mistakes like this, okay? All right. Let's talk about the macrolide group, still in protein synthesis here. That's what we're doing. So the macrolides are going to just target the 50S portion of the ribosome. You don't need to know that for the test. Sorry, unless, of course, this is one of your drugs for your paper, and then you'd want to know the target. Okay. So one of the big macrolides is erythromycin. This is usually what I was given because I had an allergy to penicillins. It's also given if a patient has microorganisms that are resistant to penicillin. Erythromycin is broad spectrum, but it does have some problems. So erythromycin will give you an upset stomach. Erythromycin also will give you those little Hershey squirts. So let me just tell you, it's difficult to teach a 3-hour lab when you are on erythromycin because you have to take those very frequent breaks and find a very hidden bathroom. I know, TMI? Sorry. All right. Sometimes patients, when they complain about the problems of erythromycin, they are given clarithromycin. Get it? Mycin. Now, clarithromycin is not in red, so you don't need to know that. But know that this is another macrolide. You know that there are other macrolides given, like clarithromycin, causes fewer GI problems, but you get a metallic taste. Okay. Whoopsie. Didn't mean to do that. Okay, another macrolide that you do need to know. Actually, if you do not know what Z-Pak is by now, all right, I don't know. You need to learn this one because this is a parent's favorite antibiotic. So Zithromax is it's called sometimes called Zithromax, but it's really azithromycin, like erythromycin, clarithromycin, commonly known as Z-Pak. So your patients will get this and say, well, okay, why is this different from penicillin? You would say, oh, because it actually stops protein synthesis in bacterial cells instead of cell wall synthesis. Okay, they're not going to ask that, but you can say you don't have to take as many pills. And also, sometimes the bacteria develop resistance against the penicillin. So then we give Z-Pak, okay? Or you could also say, well, if a patient is allergic to penicillins, we give them Z-Pak. All of those are good reasons. Okay, they tend not to be as toxic because, well, look how big it is. It tends not to get in and hurt our 50S ribosomes inside our mitochondria. Let's break for a question. Or if you need to pause, you can take a break then. Okay, which of the following is not a beta-lactam drug? Cephalosporins, ampicillin, penicillin, azithromycin, carbapenems. Okay, so it's got to be D, azithromycin, right? Cephalosporins are cephs, like K-flex. Ampicillin is a penicillin. Okay, and carbapenem. So all the other ones are indeed beta-lactam drugs, and they will all inhibit cell wall synthesis by inhibiting the PBP enzyme. PBP enzymes that are the target. Azithromycin, what we just talked about, attacks the ribosome and therefore protein synthesis. Hey, who discovered penicillin? Okay, Fred Griffith, Louis Pasteur, Robert Koch, Alexander Fleming, Robert Penicillin. You didn't choose Robert Penicillin, did you? Okay, no, Alexander Fleming, the sloppy but very smart Scottish scientist. Okay. Which inhibits bacterial cell wall synthesis? Okay, think about what came first. Penicillins, ampicillin, vancomycin, bacitracin, or all of the above? Yes, all of the above. The first two are beta-lactam drugs, the penicillins, but vanc is different and bacitracin is different, yet all of them do inhibit bacterial cell wall synthesis, all of the above. How about another class of drugs? These are the folic acid or folate synthesis inhibitors. So we call these the sulfa drug group guys, okay? So, okay, now sulfa drugs, these kinds of drugs actually can be used for things other than bacteria. We're starting to get into some gray zones here. So, but first of all, let's talk about the kinds of drugs that knock out bacterial action. Now, the sulfa drugs have pretty good selective toxicity. My example, first of all, well, it's something that blocks an enzyme it blocks an enzyme that converts paraminobenzoic acid into folate. The what? Okay, look, let's just abbreviate it. PABA. So this enzyme over here is the enzyme that is the target. What it normally does is it takes in PABA here into the active site and converts it into folate. But the sulfa drug, like sulfonamides, gets in there instead. So it's a competitive inhibitor. So the reason this works so well on bacteria is because, well, this is the only way bacteria can get folic acid or folate. You and I, we can take folic acid in our pills, can't we? Yeah, through our diet, we can do that. But bacteria don't recognize folic acid. They can't do it. We can convert folic acid into folate. It's not a problem. Bacteria, not so much. So they starve. Without folate, bacterial cells can't make DNA. Without being able to replicate DNA, the bacterial cells can't replicate. And if the bacterial cells can't replicate, okay, they die. By the way, why is folate or folic acid recommended to pregnant women? Okay, so they have little cells growing, right? The fetus is growing, cells are multiplying. That means they have to replicate their DNA. They need a lot of folate or folic acid to do this. So that's why we give our patient folic acid. Okay, all right. So, good example of a drug, though, that does block the bacterial production of folate is the sulfonamides, including trimethoprim. Again, it's a competitive inhibitor, if you remember that from the enzymes. Sulfonamides is that's a big group. Okay, and there's a lot. Trimethoprim is one of the most common. Okay, this one is FYI, but you know what? There are other organisms out there that are not bacterial but can cause disease that can be inhibited by some kind of sulfa drug by stopping their folate production. So, two examples of this, and we haven't talked about them too much, but these are malaria parasites in yellow. For some reason, the parasites all have yellow. These are red blood cells, and the malaria parasite is dividing. And yeah, so you can use certain kinds of sulfa drugs, sulfadiazine, to stop the production of malaria. Also, if a patient has toxoplasmosis, which is a protozoan, a eukaryotic protozoan, it will work on them. So these are anti-protozoan drugs. You don't need to know. These are FYI. All right. Let's talk about nucleic acid synthesis inhibitors, the fluoroquinolones. Oh, yeah, I remember those. She talked about fluoroquinolones. It's a terribly long name, but think of flowers. Okay. Actually, they're too toxic to think of flowers. So sometimes doctors will abbreviate this as an FQ because fluoroquinolone, yeah, that can be tough to say. I understand. Fluoroquinolones are completely synthetic. They discovered these in the lab when they were trying to find a way to kill malaria parasites. I know, back to malaria. Okay, what does it target? It targets this thing, the the topoisomerase enzyme, also known as gyrase. Now we have these as well, but the bacteria have unique types of gyrase topoisomerases, okay? So if you saw topoisomerase or gyrase on a test, that is the target. If you're doing your paper on a fluoroquinolone, just say the target is DNA gyrase or topoisomerase. I'm okay with that. So, okay, if the drug stops this, what happens to DNA production? Well, DNA can't replicate. And when it tries, well, DNA will break. Why? Because topoisomerase's normal function is to help the uncoiling and reattachment of parts of DNA as it untwists. See, this is a ladder all twisted. When it becomes untwisted, necessary for DNA replication, okay, it breaks, but topoisomerase stops the breaking. Block topoisomerase? Well, the bacterial DNA will shatter, break in lots of pieces. Fluoroquinolones do come in generations. If you remember, we'll talk about two of them that are just part of one generation. But the first generation, not in red, is norfloxacin, so you don't need to know that. We don't give a lot of norfloxacin, I guess, anymore. Second generation is Cipro and Levaquin. Ciprofloxacin, Levaquin. Cipro only useful for gram negatives, Levaquin for gram positives. By the way, these drugs, the fluoroquinolones, are pretty toxic drugs. They are, for reasons we are just barely starting to understand. Doggone it. Sorry about my cell phone keeps becoming annoying or being annoying. Sorry. Um, Levaquin, especially, we know deposits in the bones, and they were able to say, we can't give this to kids ever. Because no kitty vinkies, no kids, we don't see really resistance. Now, if you're wondering why this sweet little girl has her finger up her nose, I want to mention that little children, these adorable little things, and they are treasures, they are little incubators. They get a lot of germs and they get a lot of infections and they get a lot of antibiotics. The antibiotics and the bacteria that are inside the little adorable little germ factories, those antibiotics will select for resistant forms of bacteria. So kids, yeah, sorry, they're resistant. They are germ factories and they are resistant germ factories. That's all there is to it. And because we can't give Levaquin to kids, we haven't really seen resistance so far. If you're wondering where I'm getting this information, if you're angry at me, again, this is a Ken Thompson from Creighton University. He is a medical microbiologist and he teaches for the, the pharmacy department. Okay, or at least he did. Sorry, he has semi-retired. He fell in love, got married. Okay, third generation moxifloxacin. Again, you don't need to know this. It's not in red, but I do like saying moxifloxacin, so I've included it. All right. Problems. Okay, understand these drugs, when you need them, you got to take them. However, well, for good example, when the anthrax outbreak hit, we gave Levaquin and other fluoroquinolones to those people, but some people actually ended up on dialysis and got very ill. These have lots of problems. And like I said, there is resistance against Cipro. Sorry, that wasn't what I wanted to come up first. But toxicity issues are bad. They can cause cardiovascular as well as nervous system problems. Um, People who have taken fluoroquinolones for Lyme disease have had some really bad outcomes. Okay, just so you know. All you have to do is look online, Google it. You will see some angry bloggers out there. Let's talk about quinolones. They sound like fluoroquinolones, don't they? Yeah, they're related to fluoroquinolones. They were made, again, in the laboratory. The ones that we use are made in the laboratory. These quinolone drugs that I'm going to talk about now actually work on protozoans. Again, we're back to malaria. The reason this is significant is because some of your patients will be traveling to areas that have malaria. Malaria is common throughout the world, wherever it's kind of warm and humid. If there's a rainforest, there's probably a mosquito that will transmit malaria. Again, the same effect, it's still the same. Quinolones, like fluoroquinolones, are going to affect the enzyme that uncoils DNA during replication. The drug that I want you to know about is mefloquine. So back to that, remember I said I was at a traveler's clinic when I was going to Ecuador for the first time. And Dr. Marvin Bittner, who I've known for years from research, was running the travelers clinic. I'm like, hey, Marv, how's it going? He's like, how's, yeah, fine. Marv is extremely smart, really nice guy. And so he's going through the list and he's asking me all these questions and including, so, okay, I need to give you an anti-malarial medicine. So I need to find out what'll work. He said, do you have any psychotic disorders? I said, yeah, I can't take mefloquine. Now, if you're wondering which psychotic disorder your teacher has, or at least I used to be a sleepwalker. I don't know if I still am. But my dear friend went to Africa years before we all went to Ecuador. And he doesn't normally sleepwalk, but he was on mefloquine and he thought a lion was attacking the tent and started to pull the poles out from the tent to fight off the non-existent lion. So understand that this is used for malaria, but it does have some consequences. Shout out to crazy Stephen King movies. Okay. Metronidazole, Metro, okay? Metro is used. Sometimes people will say Flagyl. It is so much easier to say Flagyl, I know. This has various uses, but I really don't want to go into much detail about it. It's really good against attacking Giardia that causes giardiasis. So protozoans with flagella, they have flagella, so they get Flagyl. And yeah, so this will work on Giardia that causes giardiasis, and its cousin, Trichomonas vaginalis, that is sexually transmitted or not. There are some forms that you are born with, you get it from your mom, trick. So, Flagyl is also used sometimes when patients have stomach ulcers. Okay, so if you have Campylobacter or you have Helicobacter pylori, then sometimes Flagyl is prescribed. But I'll tell you what, this messes with these bacteria. It only works for anaerobic cells that have anaerobic metabolisms, but it will kill those bacteria, causing the DNA to break for different reasons than the fluoroquinolones. I just wanted to show you here, just it shows breaking DNA. That's all you need to know. Okay. Oh, by the way, if you have puppies or kittens that come home and they have a lot of diarrhea, chances are the doctor's going to give them Flagyl. And that's what it is. They're probably looking at maybe protozoan parasites. Okay. Cell membrane disruptors. Okay, so we're moving in. Okay, so some of the advantages of cell membrane disruptors. Number one, the microbes don't have to be actively dividing, so that's a good thing, I guess. It does bind pretty well to cell membrane phospholipids, so if there's a membrane there, they kind of like it. It does ruin selective permeability and causes the cell to lyse. Disadvantages. All cells have membranes. We have membranes. Your cells, brain cells, heart cells, liver cells, kidney cells, the neurons, they all have membranes. Now, these drugs will attach to the pathogen's membranes more to your membranes, but it's difficult. So again, the drugs target their membranes more, quote unquote. So what we do, say again, so this is what do we turn to if our gram-negative pathogen can't be killed with any other drug, or then we tend to turn to cell membrane disruptors. Remember I talked about carbapenem resistance, and if we can't give a beta-lactam drug or anything else, a lot of times we go to these drugs. These can be very toxic. We also sometimes have to use these drugs for fungal infections, but fungal organisms are eukaryotic cells, cellular creatures, and so are we. So again, there's too many similarities and there's a lot of toxicity problems. Um, unless you can do something topically, but a lot of these drugs are not. So again, some of these drugs are so toxic, we took them off the shelves until we had to bring them back. This is just showing you what some of the drugs will do. They attach to cell membranes and cause them to break. Again, they usually attach to structures that are more bacterial than human. More, quote unquote. Here again, Pseudomonas aeruginosa, gram-negative bacterium. If this is in your patient and you haven't been able to knock it out with carbapenems or even a carbapenem combination with like Cipro, which is a fluoroquinolone, so you can do cell wall synthesis inhibition and DNA synthesis inhibition, and if that isn't working, this is it. If that doesn't work, your patient will die. Unfortunately, my brother died of Pseudomonas, and there wasn't anything they could do. There's a little bit of Pseudomonas, a little bit of C. diff, and the Pseudomonas was resistant. Okay? All right. So examples of cell membrane disruptors, polymyxin B. This guy, yes, it is injectable. Yes, this is actually, I use the image from the polymyxin B people. So it does get in and it does break the membranes. Really what it likes to attach to is that freakish outer membrane of gram negatives. It has the strongest affinity or attachment to gram negative LPS. Remember, LPS is an endotoxin. So this will work on Pseudomonas membranes, but it can be toxic to other membranes too. Human cells. So this causes nephrotoxicity and neurotoxicity when being injected. This does have cumulative toxicity as well, so you don't want to use these drugs too commonly. Unfortunately, if you're not using bacitracin, guess what you're using? Yeah. And for gram negatives, for an ointment, this is all that's out there. So don't use these drugs unless you have to. And if you can, if you just had a biopsy, okay, use polymyxin. What are you going to do? But if you can, use soap and water. If you can't, try not to put it on the wound when it's still way open and bleeding so it doesn't get into the bloodstream. Like I said, this all comes from Creighton University in the Med Micro department, but there's a link if you want to look this up as far as cumulative toxicity from Google Books. Okay, another cell membrane disruptor. This one is amphotericin B. Also, there's something like it called nystatin. This is used very commonly for systemic fungal infections. You don't need to really know that that's what it does, but like I said, usually we use this for gram negatives. These kinds of antibiotics are used for either gram negative bacteria that can't be treated any other way or for fungal infections. So again, the target is a fungal membrane structure called ergosterol. Remember that your cells have a cholesterol, like, sterols. Um, Fungal cell membranes have ergosterol. It's a little different, and the drug attaches to ergosterol a little better, a little more often. Produces a pore. So here we go. Here's the fungal membrane. Here's the ergosterol. And again, it puts a nice, so the drug amphotericin B will put a hole here. That will mess with selective permeability and will kill the cell. If you destroy a cell's permeability, the cell dies. Okay? Yeah, every time. This is commonly used for, like I said, fungal infections, pretty commonly for candidiasis as well. Okay? But the problem is if this is given orally or as an injectable, it is toxic to mammal liver cells. And so liver enzymes must be tested regularly. So must have blood monitored for liver toxicity. Okay, so let's stop for a moment. If you needed to take a break, that's great. Get some coffee. Okay, kind of relieve your bladder. Come on back. And let's have a question. Okay. So, if your patient is allergic to penicillins, which particular antimicrobial should they stay away from? I know that's ending in a preposition. Too bad. Okay. K-flex, azithromycin, tetracycline, erythromycin, or flagyl. Now, flagyl, you would probably not be a probably wouldn't be a problem. Erythromycin, tetracycline, and azithromycin. Okay, these are the same protein synthesis inhibitors. K-flex. Oh, that's a cephalosporin. Oh, yeah. So cephalosporins like K-flex is a beta-lactam drug. And again, the cephalosporins are close to penicillin. And so I don't want to take that. So yeah, your patient needs to stay away from K-flex. Okay? Okay. So which of the following is a true antibiotic by definition? Penicillin, fluoroquinolone, levofloxacin, ciprofloxacin, or they're all antibiotics by definition. Only penicillin, right? Penicillin G, that hurts, and penicillin V. Okay, fluoroquinolones include levaquin and ciprofloxacin. So, and they are all completely synthetic. Okay. All right. How about just some FYI things? Other things that target membranes. Terbinafine, you see this on the counters. This is Lamisil. If your patient has a really bad nail infection, they will probably be given Lamisil. Okay, so this is a cell membrane disruptor. Yeah, and you can't do this topically, and here's why. These kinds of fungal organisms get deep in the nail beds into the fatty tissue. So you're going to have to take this orally. And does that sound bad? Yeah. So you have to take this in small doses over several months and have your liver enzymes tested. So it can be toxic to liver cells. Not a favorite, but yeah. So you go in for that mani-pedi, and they didn't sterilize their instruments. This is what you get. Okay, here's something else. This is one of the most common types of antifungals. So this is monostat. Okay, oops, sorry, there we go. Monostat, which is actually an azole or miconazole. Anything that ends in azole is going to affect any fungal organisms. Usually we give monostat and all azoles topically somehow for candidiasis yeast infections, aka yeast infections. So this does a really weird thing. It gets in and stops the mitochondrial membrane, something called a so there's, it stops an enzyme that makes ergosterol, and that's what it does. So it's all, yeah, still messing with the membranes. Another question, yeah, I just put these in here. Doripenem is a chemotherapeutic agent that attacks cell wall synthesis and is one of the few antimicrobials that we have left to treat tough gram-negative pathogens. Based on the name alone, which group of antimicrobials does it belong to? Cephalosporins, carbapenems, penicillin, vancomycin, isoniazid. Right, doripenem, carbapenem. And again, this is a beta-lactam drug. We use it, and if it doesn't work on gram negatives, we are very scared. Okay, which of the following chemotherapeutics attacks cell wall synthesis, is chemically very large, is narrow spectrum, and should be a comma there, and only kills gram-positive bacteria, and is used to treat MRSA, MRSA? Okay, did I describe enough of it? Ampicillin, carbapenem, cephalexin, like K-flex, bacitracin, or vancomycin? Of course, vancomycin. That's the one. It's huge, toxic, narrow spectrum. Kills Staph aureus, hopefully. Inhibits bacterial protein synthesis. So now we're going to move from cell wall synthesis to the protein synthesis inhibitors. And is very broad spectrum, and it really gets into cells and tissues to kill intracellular bacteria. However, it can inhibit birth control pill efficacy efficiency, and depress the immune system. Vancomycin, tetracycline, penicillin, sulfonamides, polymyxin B. Okay, vanc is a cell wall synthesis. So tetracyclines, B, good. Yes, B, tetracyclines. Penicillin is again a cell wall synthesis, beta-lactam drug, okay. Sulfonamides, yeah, that's going to stop folic acid, polymyxin B. All right, that's a membrane inhibitor, okay. All right, so real quick, what about parasitic worms and parasites? Well, for parasitic worms, we give drugs that are called antihelmintic. And the reason it's called this is because a fancy science name for worms is a helminth. So parasitic worms are helminth. So this is like an antihelmintic. So that spelling, you will see it. It varies. It really does. I'm only going to have you look at ivermectin. It's the only one you need to know. It's part of another bigger group, but this one is commonly seen. And what it does is that it causes esophageal paralysis in the parasitic worm and the adult worms, and we're going to find that it also is used to kill insects, ectoparasites in just a minute. But the reason this works is because parasitic worms and insects have to feed all the time. They can't make fat or glycogen, so they have to eat, eat, eat. And if they don't get a meal within 48 hours, they do starve. This is only true with the adult parasites, though. If they form an egg, that's a different topic altogether. By the way, this does come from a type of bacterium called Streptomyces. So, and I have, if you work on farms, you probably know that animals get dewormed and they probably get ivermectin. Sometimes they use it on sometimes trees get worms as well. All the pines are dying. My pines had parasitic worms, so even my pine trees got a type of ivermectin. But let's say your patient comes in with an ectoparasite like mites, also known as scabies, and head lice. All right. Well, these are little insecty bugs, the real bugs. And again, the patient is usually prescribed ivermectin. Again, they're paralyzed. They can't feed off of your fluids and tissues, so they starve. Okay. I know. Ew, moment. Here's some really cool old remedies. I won't go into these, but if you want to read them, they're fine. Understand that tobacco nicotine used to be given to livestock, but yes, it might work to kill the parasite, but again, it would probably hurt the animal. So here's a cat smoking. The other things, again, some of them will work, but it might kill your animal. The other ones that don't work probably are pretty lame. There's even a wormwood that, well, they named it because it was used to treat worms. But again, if you get it to the right toxicity level where it would paralyze the worms, because they're going to have to eat a lot of it and it has to be fresh, it'll probably hurt the animal. So, nah. So I'm not going to go into these. You can look at these when you have time. All right, let's stop and talk about a super infection. All right, is this something Superman and the superheroes get? No, I know. Wah wah. Whatever. Okay. No. This takes us back to a few things, few topics. Remember I said not all bacteria and microorganisms / germs are created equal. They're different. And not all antimicrobials are going to affect everything the same way. Okay, you run the risk of destroying good guys, the normal microbiota, or leaving some behind that will overgrow and cause a superinfection. Or you kill the easy little guys to kill, and then the mutants that have developed a little mutation in one of their genes and have become resistant to an antibiotic. Okay, well, so then you get a superinfection with a superbug. Okay, so here are some of the scenarios. Forgive me, I developed these animations because I didn't have a textbook, and I thought if I'm going to do this, I might as well just go big or go home. So let's suppose your patient is prescribed an antibiotic. So here it goes. Now I'm only going to have the bacteria at first in the colon. Now understand a lot of this pertains to other parts in the body. Okay, so it's traveling through the colon, and what happens as it travels through there? Remember, antibiotics are poison. Some bacteria are going to be more resistant and some more susceptible than others. So, okay, some of the microbiota is dying, or bacteria. They could be pathogens. But look who's left. And then they divide. The ones that survive will divide and take over, causing a superinfection. And so, why would they survive when everybody else is dead? Well, okay, in this scenario that I'm going to talk about, what's the hardest thing to kill? Oh, yeah, anything with an endospore former. Guess what? You have a lot of endospore formers in the the most distal part of your colon. These are actually not quite that far down, but so Clostridium difficile. Remember, Clostridium is an endospore former. C. diff. They will be left. Everybody else is gone. Maybe some of them were causing disease. Maybe some weren't, probably a lot were not, but no matter what, the toughest guys in the world making the endospores, those little black dots, they're left. And what happens when they're left? Again, they overgrow and that can cause a terrible inflammatory response. And we say that this is pseudomembranous colitis. If you want to see what C. diff really looks like, there you go. Some endospores forming in there. And how bad can this get? Well, necrosis and a perforated distal ileum is one of those things that can happen. So this is bad. And you see the megacolon there. This is bad. So that patient's very sick, and some patients can actually die as a result of this. So very, very, very bad. By the way, we never used to have C. diff, or at least you never heard of it. In the 1980s, that's when we started to routinely put antibiotics into everything. So the pharmaceutical companies aren't going to have a problem with it because they sold an awful lot of antibiotics. It got dumped in the feed. The farmers didn't mind it because they could, and this is not their fault. It isn't. They were just trying to grow enough animals to make a profit. But if you grow animals in a small confined space, they're going to have a lot of infectious diseases, so you're going to need antibiotics. Also, farmers get paid per pound, and antibiotics make animals fatter. Well, probably because normal microbiota has been wiped out, and the ones that are left make you fat. That could possibly be it, but it's a hypothesis, you guys. Okay, so let's again say that your patient is prescribed an antibiotic and they need it. Okay, but okay, one bacterial cell develops a little mutation. It's an accident. They don't get to choose this. They had the mutation before the antibiotic even got there, just by replication mistakes, okay? All right, so what does that mean? That means that, okay, some of the bacteria is going to disappear. Who's left? Well, the antibiotic resistant bacteria, and they develop. And now you have a superinfection of superbugs. Yeah, the resistance is passed on to the offspring, and they are all resistant to that antibiotic. Superbugs. Yeah. And some of these are found in cattle. Okay, because this all happens in the cattle too, in the feed yards. They get resistant bacteria. Oh, yum. But also you can find the resistant bugs everywhere. These are not sterile. And there, and there. And now you have an explosion. And as long as you have antibiotics in the environment, you will select for the resistant kinds of bacteria. Okay, one more scenario. I know this looks busy, but here's a lady. So we're going to talk about um, And this is true with guys as well in their way. But okay, so again, your patient has to take an antibiotic. Maybe they have a urinary tract infection. So maybe I should have lifted that up a little bit further. But yeah, she has a urinary tract infection. So she takes an antibiotic. And again, this will kill bacteria, sometimes the ones you don't want to kill, but also that leaves, okay, something that isn't bacterial, yeast. In your oral cavity, in the vagina, you've got a lot of yeast, ladies. And, for the guys too, they're everywhere. Okay, they're part of your normal microbiota. They're not a problem when there's a balance. But when these guys leave, they are left, and boy, they will take over. And you get candidiasis, either thrush, oral candidiasis, or vaginal candidiasis, also known as a vaginal yeast infection. So this happens a lot with ladies. There's a lot, they take their antibiotics and maybe they needed them, maybe they didn't, but now they have yeast infections. So again, keep in mind, not all microbes are equal, and your normal microbiota is always essential for balance and health, right? Okay. All right, how about a question? This expensive antibiotic can cause red man syndrome if infused too rapidly. The expensive part pretty much was the giveaway. But okay, azithromycin, flagyls, gentamicin, vancomycin, or chloramphenicol? Vancomycin is correct. Which of the following would you use to target a fungal membrane ergosterol? So membrane, okay. Penicillin, vancomycin, chloramphenicol, amphotericin B, carbapenem. Amphotericin B. And again, it is going to have some toxicity problems. Erythromycin effects. Okay. Nucleic acid synthesis, protein synthesis, plasma membrane permeability, cell wall synthesis, or all of the above. Well, that's a macrolide, and so it's not a cell wall synthesis. That was the next group, which were protein synthesis inhibitors. So B, yes. Okay. All right. Oh, we're back to this. Okay. So imagine a world without antimicrobial medications. My grandmother used to flip out when she heard the word strep throat. You hear the word strep throat like your child has strep throat, and you probably think, well, I just run out and get an antibiotic. But remember, okay, antibiotics weren't really readily available until the 40s. So for my aunt, the prognosis for a group A strep infection, Streptococcus pyogenes, probably, it was very likely a death sentence in babies and young children. Um, so this was bad. Think way back to Semmelweis. That was strep, and even some staph infections could be lethal. So what do we do when the meds aren't working anymore? Okay, so we have a lot of problems these days. There's a lot of misuse of antibiotics, and that's coupled with the way microbes normally evolve. They do. If they're going to divide and divide quickly, they're going to mutate quickly. Some of those mutations are, all mutations are random. Some of those mutations will be useful to bacteria, some harmful, some might not make any difference at all. But they've got numbers and they've got mutations on their side. So because of all that, resistance threatens our abilities to use these medications. Current bacterial resistance is causing complete clinical failure of everything, and then we have nothing. So how does resistance occur? So random mutations cause resistance. Bacteria become magically resistant to these poisons. How? Okay, so what's with the little Pac-Man? Okay, these are enzymes. Okay, picture these if you will. These are little enzymes that bacteria can make, the little yellow Pac-Men are enzymes that bacterial can make and then release into their environment. And if a little white pill and like the penicillin poison gets into their environment, well, these enzymes will destroy these little pills, the penicillins, before the drug gets to the bacterial cell. Okay? So we call these enzymes that bacteria make beta lactamases if we're talking about just in general. So if the bacteria are making enzymes that knock out most of these drugs, okay, we call them beta lactamases. Sometimes they're specific. Penicillinases knock out the penicillins. This is pretty bad. A type of MRSA does this, but the MRSA has other ways of being resistant. The bacteria then also developed a mutation to form little cephalosporinases, so they're resistant against cephalosporin drugs. That's really bad. And usually the bacteria that can make penicillinases also make cephalosporinases too. So, or and there are also those bacteria that can make carbapenemases. These again tend to be enzymes that wipe out our very, very favorite drugs, the carbapenems. Usually the bacteria that can make a carbapenemase can also make a cephalosporinase, can also make a penicillinase. So basically they're just resistant to all the beta lactams. So they're making it in general carbapenemases, beta lactamases. So, and you don't need this for the test, but if you ever see KPC or NDM on a chart, know that things are really bad, and you'll probably see the panicked look in the ID infectious disease MDs in their face. It's bad. So if you can't quite picture what's going on, so here we have our bacteria, right? Here they are dividing, and the PBPs are busy linking together the peptidoglycan, and they're all happy. But let's send in the poison. Let's send in the penicillins and the beta lactam drugs. But the bacteria secrete and make little beta lactamases. So the drug can't get to the bacteria because the little penicillinases are knocking them out. This is just one method to be resistant against antibiotics. There are other ways, and again, bacteria can do this by spontaneous mutations. They can also acquire new genes from other species. So, for example, Enterococci bacteria developed resistance genes against vancomycin, producing vancomycin-resistant enterococci, so VRE. Over here, these guys normally live in your gut. This is Enterococcus faecium, enterococci. They developed the resistance to vancomycin. They have the gene and they became vancomycin resistant. Now, they gave these genes to other bacteria like Staphylococcus aureus, making vancomycin-resistant Staphylococcus aureus. This one's easy to remember because scientists called it Visa. Now, the Visa Cup people hated this, and they tried to change the name. We still call them Visas. So this is pretty bad, but there are a few other drugs that we can give, okay? Also, Streptococcus pneumoniae can also get this gene from Enterococcus. So again, Enterococci were the first to get the gene, and so they became VRE. They gave the gene to Staph aureus. Staph aureus became Visa and vancomycin resistant. And then it also got to strep. And I'm not going to have you learn what the resistant strep pneumonia is. All right. So some of the things that can happen, I just want to show you. These are all the ways that bacteria can be resistant to antibiotics. So one bacterial cell can have a plasmid with lots of genes. Some of the genes will make penicillinases, but other genes will make little pumps so that the antibiotic gets into the bacterial cell. They spit them out. You don't need to know that. Sometimes they change certain structures, like they'll alter their PBP. They'll develop alternative pathways, but you don't need to know any of these. Just know what a penicillinase is, okay, or a beta lactamase in general. Okay, these guys. All right. All righty. So just again, how did this happen? How did bacterial resistance evolve? Well, here's our beta-lactam drug, ampicillin with a beta-lactam ring. The enzyme, our Pac-Man, can cut that ring and then it no longer works, broken ampicillin. So originally you had a whole bunch of bacteria, and again, they just naturally divide, and when they divide, they naturally mutate, and some mutations have no effect. Some are harmful to the bacteria, but in the right environment, a mutation will make a bacterial cell the strongest, the most fit. Remember Darwin, survival of the fittest, descent with modification. So originally, let's say these bacteria originally had just a normal gene, a little section of DNA with A's and T's and G's and C's, and that gene was transcribed and translated into an enzyme with a little active site, and this gene was supposed to take care of trash. Okay, that's all it was supposed to do. But then a little mutant came about. A little cell here got a little mutation accidentally. Maybe some part of DNA, got messed up with, like a base substitution mutation. So, or just a mutation, that's all you need to know. And so that's going to change the enzyme that's made. And the enzyme now looks like this. Okay. And now the enzyme doesn't destroy waste, but it destroys beta-lactam drugs. Voila. All right. Keep in mind, okay, when it comes to antibiotic resistance, bacteria again do not develop resistance by just recognizing that there's a threat in their environment. Okay. They don't see the antibiotic and go, oh, there's an antibiotic. I will choose a specific mutation now to make an enzyme. No, it's an accident. And they've been doing this. Remember that the penicillin normally came from the bread mold penicillium. So that antibiotic had been in nature for a while, and the bacteria had resistance genes for a while. Okay, before Alexander Fleming even figured out penicillin. Okay, but the more we have antibiotics in our environment, the more we will just select for the antibiotic strains of bacteria. So again, here's the original bacterial cell. It divides. Some of its offspring will have little mutations. If these mutations just happen to allow it to make something like a penicillinase, it will survive. These will be the only cells that survive in an environment of penicillin. If there's no penicillin, these guys are not selected for. Okay? But since these are the only ones left, these are the ones that divide and thrive and are causing global antibiotic resistance and antibiotic failure. Okay? So what do we do? Stop giving antibiotics? That would do it. That would do it. But do we want to go back to the Victorian era where maybe you lived to the age of 40? Don't think so. That's if you're lucky. And again, without antibiotics, you couldn't have any surgical procedures, no transplants, nothing. Even dental procedures would be iffy at best. So maybe if we're just better stewards about how we use antibiotics. Okay, so let's change gears and talk about non-living things. How about the antivirals? Yeah, we touched on this with HIV. Keep in mind there aren't a lot that are available and many are toxic. See the notes about AZT. So selective toxicity is hard because the viruses use our cell and our cell stuff and our enzymes and all of that for manufacturing new viruses. CAZT notes. There are three typical strategies that we use to fight viruses. Number one, cause mutations in the faster replicating DNA, which would be the viral DNA. That's going to be replicating faster, so we could hurt them more. Number two, target unique viral structures if we can find them. 3, manipulate the host's immune response to recognize the invading virus. Okay, one at a time. All right, how about influenza? So I'm going to talk about the development of influenza drugs. Mind you, this is not a vaccine. Vaccines train your immune system. Okay, so you do all the fighting. You don't have to take a poison. All right, you have an immune system set up. Vaccines just help your immune system be a little smarter and faster. Okay, so these are poisons, these are drugs to stop the replication of viruses. Now, for influenza viruses, there were some drugs that were developed. There was a drug called amantadine, but the viruses developed very rapid resistance. Ramanadine, also rapid viral resistance. Remember, these guys replicate fast. Then there was a drug group called zanamivir, and it was approved for treatment of persons who were older than 7 years. And this attacked an enzyme that influenza viruses need called neuraminidase. So this drug blocked neuraminidase. It was a neuraminidase inhibitor, and it was used for clinical use and used for influenza A and B. But what about for younger kids? Well, that's when the next one came up. So this group of drugs, they're called the oseltamivirs. I know. If you remember Tamiflu, you'll probably be fine. Um, it was approved for treatment of persons that were at least a year old or older. So if you're less than a year old, you can't have it. But what you're going to know is Tamiflu. Tamiflu is oseltamivir. Generics will probably have that. This is something called a influenza virus neuraminidase inhibitor, and I'll show you what that means in a minute. Drawbacks to this: it has neuropsychiatric events. Some of you might have observed this in patients. So a little more. So again, a neuraminidase inhibitor. Over here is the influenza virus to the right. And you can see there's these little yellow spikes, okay? So the yellow spikes is hemagglutinin, but then there's a green spike over on the lower right-hand side here, looking. Okay, little red circle with a green stalk. This is an enzyme protein called neuraminidase. Neuraminidase allows these viruses to escape from host mucus, okay? Without this, without the neuraminidase, technically the virus has a really hard time escaping your mucus, and therefore it has a hard time infecting the next cell. Okay, Tamiflu inhibits this. So technically, again, the virus is supposed to be then stuck in the mucus, and the mucus can carry it all away. All right. So here's some problems with Tamiflu. It's no good unless you can start your treatment within 24 hours of symptoms. So you get up Monday morning and I'm achy, maybe a slight fever, I'm tired, kind of a dry cough. Man, I'm coming down with a cold again. 3 days later, you realize you're really bad. You go into the doctor, and the doctor says, you have influenza. And you say, can I have Tamiflu? I heard about Tamiflu. I don't want to have it anymore. And a good doctor will tell you, no, Tamiflu has no effect unless we caught this within 24 hours of symptoms, meaning you would have had to have started to take it by by the end of Monday. But yeah, Tuesday afternoon would be way too late. So again, sadly enough, people think they have a cold and they do not get they don't get the neuraminidase in time. Especially for older people, there are some bad problems that can cause neuropsychiatric events. But some physicians will still give it because now patients know that you can't give antibiotics. And so, but the patient wants something, and the doctor's like, well, if I give an antibiotic that has no effect, I don't know, maybe Tamiflu might help. But no, it won't. A good doctor will say, I don't have anything for you. It's been over 24 hours. So go home, get some rest, drink lots of fluids, get some juices, colorful foods, go watch some Pixar cartoons and just do the best you can. Okay. Okay. Another antiviral. Let's attack herpes viruses. So, these drugs that usually are used to attack herpes tend to be nucleic acid analogs. Oh, yeah, like AZT. Sort of. But this one is a little different. It's not as toxic as AZT. Acyclovir um, does look like the DNA guanine. So remember, G's with C's over here. Looks like guanine. So his DNA is being replicated. The enzyme that kind of matches everything up accidentally will put a cyclovir there instead of guanosine. Okay, they look the same. They're base analogs. Okay, and that will cause mutations in the developing herpes viruses. So when we're talking about oral herpes, genital herpes, chickenpox, shingles, all of that. The reason this is not as nasty as AZT is that, well, there's a lot of interesting things that have to happen to acyclovir, and the virus actually screws up and makes acyclovir in the cell, makes the deadly form. So it's kind of interesting. So it's hard to explain, but just know it's not as nasty as AZT, okay? But still, you don't want to abuse it. So I would, I'm not crazy about any antivirals, but Abreva is a cyclovir that you put on just topically. So if you get one of these, you can just add Abreva, and it's acyclovir topically. And again, it will mutate the viral DNA. More than it'll hurt ours. So there you go. The last one, sometimes when your patient has hepatitis due to hepatitis, usually B or C, we really don't have a lot to give them. There is a new class of drugs that are extraordinarily expensive, and if you have $1,000 per pill, and you're going to have to take them for 6 months, you can get treated for hepatitis C, but few people can afford that. So the only option is the old option, and that's interferon. Interferon, quote unquote, interferes with viral replication. So, what does interferon do? It interferes with viral replication inside the host cell. Typically, interferon is prescribed to patients with the hepatitis viral disease. Interferon is a chemical that is normally made by cells. Interferon is an emergency chemical signal, kind of like a hormone, but it's made by many cells, like white blood cells. They're made by your cells when there is a viral infection. Now, the cell that makes the signal is a goner, but it can send it out to the other cells. Now, sometimes patients will get a viral infection, but they're not making enough interferon, so the drug is prescribed. What does it do? It shuts down cell manufacturing processes. So if the cell isn't manufacturing anything, then the virus can't be manufactured either, right? But it's very toxic. Took years. We've been tweaking it forever. There are many types of interferon. Many patients will tell you that they wouldn't have even gone through therapy except for the love of their family and friends, loved ones in general, because it's horrible. I guess they say the only way you can think of this is if you had the worst case of influenza nonstop for months. That's what it's like to take interferon. But the option is probably death, right? So there is another method though. They're called interferon inducers, and they make the body think that they actually trick the body into making more of its own interferon. It makes the body think, oh, recognize that there's viruses around, is what it does. I won't go into the details, but that's what it does. Let's do some questions. Which of the following drugs is a DNA analog for guanine, guanosine actually, and is used to treat herpes viruses? AZT, acyclovir, penicillin, Tamiflu, or interferon? Acyclovir is correct. Okay. So, which of the following drugs is prescribed to treat influenza, but is essentially worthless unless treatment begins within 24 hours of the onset of symptoms? AZT, acyclovir, penicillin, Tamiflu, or interferon? Right, D, Tamiflu. It's not going to work. It's not. Okay. This very toxic chemical interferes with the replication of hepatitis viruses. AZT, acyclovir, penicillin, Tamiflu, interferon. Interferon. Interferon interferes with viral replication. So what would penicillin do to any of those viruses? Again, nothing. Penicillin, the antibiotics, only attack things that bacteria have, right? Only things that bacteria have. Viruses have a protein capsid, they have nucleic acid, and maybe some unique enzymes to attack. That's it. Okay, precautions when you are taking antibiotics. We need these. We need to be good stewards and we need to teach other people. You need to teach other people. I'm only going to be your teacher for a few more weeks. So you need to go forth and be good stewards of antibiotics, okay? So, number one, vaccinate if possible. But Sally, we can't vaccinate for every infectious organism. So, and for reasons that I'll cover next week, okay? But if you can vaccinate, don't use antibiotics unless absolutely necessary. Be good stewards, okay? Do we really need to put all these antibiotics into feed? Okay, they stopped it in Europe. Now, mind you, again, it's not the farmer's fault. They're trying to make a living and they barely make a profit. But we just need to rethink the way we do things. Myself, now, when the time comes, maybe you can afford to get antibiotic-free meat, okay? But right now, I know you're paying debts. Use the correct dosage to overcome the infection quickly. Do you remember some antibiotics do not kill? Remember, they're the protein synthesis inhibitors like Z-Pak that will go in and they will stop ribosomes. They block them. The bacterial cell isn't dead. So what if you didn't take enough antibiotic to block every single ribosome in every single bacterial cell? Well, they'll have other ribosomes to make proteins. And they're not dead. So they can always make more ribosomes. So, and they'll be fine. You won't be fine. They'll be fine. Never take someone else's prescription. Remember chlorphenicol. Take the complete prescribed amount, even if you feel better. Symptoms are almost always the product of the inflammatory response. It's your immune response. When you feel better, it's probably because you aren't responding as violently to the invasion. Just because you don't have a fever or aches or anything like that doesn't mean the bacteria still aren't hiding in the crevices of alveoli, hiding in biofilm, all over your body. So please take the complete prescribed amount. Also look at point number 3. Change the antibiotics at the first sign of resistance. So when do we know it's not working? And again, the antibiotics may not be working for many reasons. Maybe the drug is narrow spectrum and it knocks out gram positives, but you have a gram negative. All right. Or maybe the bacteria in your body have developed a mutant gene and they're resistant. Okay. So what normally would have worked on them doesn't. So you had Staph aureus, and regular Staph aureus penicillin would have been fine, but one of your Staph aureus bacterial cells developed a mutation. Now penicillin doesn't work. Okay. Give your patient 3 days, 3 full days. Okay. If the symptoms are not decreasing in severity, okay, then get on the horn and call the doctor, the nurse practitioner, the physician assistant, okay? Tell them, and they'll change antibiotics, okay? Use combinations. You know, there are a lot of, and I'm going to use the B word, there's a lot of bugs that are multi-drug bugs. So, for example, Pseudomonas aeruginosa is an combination antibiotic bug. Tuberculosis definitely is. Pseudomonas, maybe not, unless your patient has, you know, uh, oh, certain conditions and you suspect resistance. But um, tuberculosis is always a combination. Three drugs to start with, and then down to two because the third one's so toxic. Okay. All right. So antibiotic resistance. So if a patient is told that they have antibiotic resistance or something's going on and they hear the word antibiotic resistance, how do you explain this to a patient? Well, when you take antibiotics and you are not good, taking them correctly, then you develop resistance. No, wrong. Nope. Nope, that's not how antibiotic resistance is occurring. It's not about you at all. The bacteria develop resistance. Bacteria have resistance. They always have, and they always will. And the antibiotics select for antibiotic resistant strains. That's what they do. So don't tell the patient. It's true when we abuse antibiotics, that selects for the when we abuse antibiotics, that selects for the antibiotic resistant strains. That's what it does. I know. Now, you can be resistant to sleep aids because sleep aids affect you. Okay. But but no, this is all about bacteria and bacterial cells. It's not about you. Antibiotic resistance, not about you. Hey, let's ask some questions. Hey, speaking of resistance. MRSA stands for metronidazole resistant Streptococcus pneumoniae. B. Methicillin-resistant Staphylococcus aureus. C. Metronidazole resting Staphylococcus aureus. D. Mitochondrion relay Saccharomyces aureus. E. Mitochondrion-resistant Streptococcus pneumoniae. Okay. Yes, B. You knew that. Methicillin-resistant Staph aureus. So what is this organism going to be resistant to? Probably more than methicillin. This should really be multiple resistant Staph aureus. So then what do we give the patient? Vancomycin. If not that, there are some others, hopefully, if it's not too late. If you treat a eukaryotic protozoan like Giardia or Trichomonas, which of the following would be the best agent? Remember, they have flagella. So, metronidazole or flagyl, vancomycin, tetracycline, penicillin, erythromycin. Yes, of course. Metro. Flagyl is what we use for Giardia or Trick. Okay? Yeah, it breaks up the DNA. All right. Which antimicrobial causes photosensitivity, tooth discoloration? Could be found in Kentucky Fried Chicken or any other chicken raised in the U.S. and should not be given to pregnant women or developing children. Penicillin, tetracycline, chloramphenicol, erythromycin, or ciprofloxacin. B, tetracycline is correct. Good. Which of the following inhibits DNA synthesis and can be pretty toxic? Cephalexin, chloramphenicol, tetracycline, erythromycin, ciprofloxacin. Cipro, right? Yeah. Okay, Cipro. This class or type of drugs has the best selective toxicity. Fluoroquinolones, chloramphenicol, beta-lactams, macrolides, polymyxin B. Of course, our nice little beta-lactam drugs. Yep. Fluoroquinolones, toxic. Chloramphenicol, yeah, remember destroys bone marrow in 1 in 20,000, and polymyxin is a membrane uh, permeability inhibitor. A 60-year-old female presents with a standard Staph aureus gram-positive respiratory infection. Which would be the best antimicrobial? Penicillin, anything broad spectrum, chloramphenicol, gentamicin, bacitracin. What are you going to give her? Penicillin, absolutely pound for pound. If it's just regular staph, not resistant, and she doesn't have any allergies, it is the best. Broad spectrum would wipe out her normal microbiota and she'd end up with a yeast infection. Chloramphenicol, she might end up with missing the ability to make blood cells. Gentamicin, she might end up with hearing loss or without kidneys and be on dialysis. And bacitracin, that's never given orally. So this drug is ototoxic, can cause deafness and nephrotoxicity. Basically, it can put you on dialysis. Which one was that? Carbapenem, K-Flex, ampicillin, gentamicin, erythromycin. Well, the first three are nice little beta-lactam drugs, not them. Gentamicin. Erythromycin is another protein synthesis inhibitor, but it's not as nasty as gentamicin. But again, if you need gentamicin, you take gentamicin. Hopefully you don't have to take it till later on in life. Another shout out to infection control nurses. Again, you guys, some of you might be this person. You need to go out and tell patients, well, you have to tell you need to tell staff and other nurses especially, you know, wash your hands, use universal precautions, protect your patients. Okay? Little Darwin. Remember, the bacteria are always going to mutate faster than we can get new antibiotics. But because they can mutate very quickly, it's not because they're strong. It's not because they're intelligent. They're not thinking this out. They just mutate very, very quickly, and they are the most responsive to change. And that's what makes them the strongest. Have a good one.